So, welcome to lecture 16. And so far, we have been talking about the metabolism of carbohydrates. And today, we turn a new leaf and move into lipid metabolism. Now, this lecture is divided into two parts. One is the classification of fatty acids, and this particular part should have been dealt in organic chemistry. And if you are well versed with this part, you can skip directly to the process of lipolysis and beta oxidation. And the second part of the lecture focuses primarily on beta oxidation, but we will talk a bit about lipolysis, because without talking about lipolysis, we cannot really understand the process of beta oxidation. So this lecture will follow the following roadmap. First, we will try to define what are lipids. Then we will classify lipids according to various criteria. We will focus a bit more on omega classification. So these particular points, these 3 points, are part of the organic chemistry lectures on lipids. And so if you are well versed with these parts, you can directly start with lipolysis. In this lecture, of course, we will talk about the basics of lipolysis. We will not talk about the transport of lipids by lipoproteins, because that particular part should be covered under lipoprotein metabolism, which is a part of the cardiovascular system course in the curriculum. After that, we will talk about the fate of glycerol. And once we have understood the fate of glycerol, we talk about the transport of fatty acids into the mitochondria. And the mitochondria is the site for beta oxidation. And we move directly after that into the process of beta oxidation. We have to look into the individual steps. Following that, we focus on specific calculations regarding beta oxidation, and we calculate the number of ATP molecules that are used in the process and the total and net gain of ATP from the process of beta oxidation. Towards the end of the lecture, I will introduce you to fatty acid oxidation disorders. As I mentioned here, I will just introduce you to the topic on the LMS. There is a review article that talks about fatty acid oxidation disorders. And concerning that, there is a take-home assignment. Please take that assignment seriously, because you will be expecting some SAQs from that assignment in the end of course assessment. So that's the complete roadmap for this particular lecture. So let's start with the definition of lipids. Very simply, lipids are a heterogeneous group of compounds including fats, oils, steroids, waxes, and associated or related compounds. Now, all lipids, they have carbon, hydrogen, and oxygen. And unlike carbohydrates, where the ratio of hydrogen to oxygen is 2:1, in lipids, the hydrogen to oxygen ratio is not 2:1. So that's the difference between lipids and carbohydrates, if you want to distinguish between them based on their molecular composition. All lipids are nonpolar. Since they are nonpolar, so they are relatively insoluble in water, and this is one of the reasons when you pour a drop of oil in a glass of water, you will see that the oil will be floating on top of the water meniscus. So they are relatively insoluble in water because they are nonpolar compounds. And remember that we talked about like dissolves like when we talked about solubility. So they are soluble in nonpolar solvents such as ether and chloroform. And these two reagents therefore are used for the extraction of lipids from plant or animal tissue. So coming back, lipids are a heterogeneous group of compounds which include fats, oils, steroids, waxes, and other associated compounds. They are nonpolar and therefore they are insoluble in water, but soluble in nonpolar solvents such as ether and chloroform. Now, if somebody asks you what is the type of interaction existing in lipid molecules, since they are nonpolar compounds, therefore the interaction that is predominantly present within lipids are hydrophobic interaction. If I want to classify lipids broadly, I can classify them into simple or complex lipids. Simple lipids are further classified into fats and waxes. Now, fats are esters of fatty acid with glycerol, and we will look at this particular molecules in more detail as we talk about lipolysis. Glycerol is a trihydric alcohol, and fatty acids react with glycerol to form a ester linkage, and oils are fats which are in the liquid state. Waxes, on the other hand, are esters of fatty acids with higher molecular weight with monohydric alcohol. So unlike glycerol, in this case we will have a monohydric alcohol that will form an ester linkage with a fatty acid, generally of high molecular weight. Next we come to complex lipids. Complex lipids are further classified into phospholipids, glycolipids, and there is a certain class that we call other complex lipids, and within this class we have the most important complex lipids, which are known as the lipoproteins. So what are phospholipids? So in phospholipids, in addition to fatty acids and alcohol, there is a phosphoric acid residue. You already know what phosphoric acid is, so I'm not going to dwell on that. But phospholipids also have nitrogen containing bases and other substituents, and a classical example of a phospholipid is a glycerophospholipid, where the alcohol is glycerol, whereas we have certain other kinds of phospholipids known as sphingolipids or sphingophospholipids, where the alcohol instead of glycerol is sphingosine. And these kinds of phospholipids are extensively present in the brain. Glycolipids, sometimes also known as glycosphingolipids, are lipids containing fatty acid. The alcohol is sphingosine, and along with fatty acid and sphingosine, they also contain carbohydrate. In the other complex lipid group, we have the sulfolipids and the aminolipids, but the most important are the lipoproteins, which are responsible for the transport of lipids between various organs inside the body. You may have already heard about LDL and HDL. So LDL stands for low density lipoprotein, HDL stands for high density lipoprotein. Other than that, there are chylomicrons, very low density lipoproteins, and other classes of lipoproteins which are responsible for maintaining the homeostasis in terms of lipid concentrations in the plasma environment. However, this particular topic of lipoproteins requires further discussion and will be dealt in detail in lipoprotein metabolism when you study the cardiovascular system. The third class, often classified as precursor or derived lipids, and these include fatty acids, glycerols, steroids, and other alcohols. Apart from that, we have fatty aldehyde, and one of the most important precursor and derived lipids are the ketone bodies, because they are directly derived from fatty acids, as you already know, and we will be looking at ketone body metabolism in the following lecture. Apart from that, we have lipid soluble vitamins. Vitamins such as A, E, D, and K are also derived from lipids. And lastly, we have the hormones, which are also derived from lipids, especially the steroid hormones like testosterone, progesterone, etc. So that's a very, very broad classification of lipids into three classes: simple lipids, complex lipids, precursor or derived lipids. So we move on and we look at further classification of lipids, but we will be focusing mostly on the classification of fatty acids. Now, fatty acids can be classified according to length. That means the number of carbon atoms that are present in the fatty acid, and based on the degree of saturation that is present in the fatty acid. So let's first start with classification of fatty acids according to length, or the number of carbon atoms present in the fatty acids. Based on chain length, we have three major categories: short chain fatty acid, medium chain fatty acid, and long chain fatty acid. Short chain fatty acids contain 6 or less than 6 carbons. Medium chain fatty acids are fatty acids that contain between 6 and 12 carbons. And long chain fatty acids contain 14 or more than 14 carbons. Now, if you look at the classification, you will see that all the numbers that are stated in this particular part of the slide are even numbers. That's because most naturally occurring fatty acids contain even number of carbons in their chains. Now we move to the next classification based on saturation. According to saturation, we can classify fatty acids into three classes: saturated fatty acid, monounsaturated fatty acid, polyunsaturated fatty acid. Saturated fatty acids are those fatty acids that do not contain any double or triple bonds. So they are fully loaded with hydrogen atoms, and they contain only single bonds between or within the carbon atoms that are present in the fatty acid. We will shortly look at some examples, and that will clarify the concept better. Then we come to monounsaturated fatty acid. Mono means one. So this particular group of fatty acid contain one double bond. So monounsaturated fatty acid have carbons without hydrogen, and they form a double bond between the carbons. Monounsaturated fatty acids are often termed as MUFA, and they have a role to play in the physiological homeostasis, and we shall look at them when we talk about lipoprotein and lipid metabolism in cardiovascular system. Then we come to polyunsaturated fatty acid, or PUFA, which contain more than one carbon-to-carbon double bond, and they are classified further by the position of double bond nearest to the methylene. So that's the classification of fatty acids according to the level of saturation. Let us look at some specific examples to clarify this concept better. So let us look at first a saturated fatty acid. Saturated fatty acid means there will be only single bonds present. An example of such a fatty acid is stearic acid. And if you look at the molecular architecture of stearic acid, you will see that all the carbon-carbon bonds are single bonds, and there are no double bonds present within this particular molecule. This is the carboxylic acid group. This is the alpha carbon, and this particular carbon is known as the methyl carbon, or as we shall see later in the lecture, it is also known as the omega carbon. So in saturated fatty acid, there are no double bonds present. The only double bond that you see here is in the functional group of the fatty acid, or the carboxylic acid group. We move on to the monounsaturated fatty acid group. A classical example, oleic acid. Mono means one, so there will be one unsaturation, that means one double bond present in this fatty acid. If I look at the molecular architecture of oleic acid, you will see that there is one double bond present between the carbons at this particular position. Again, this is the functional group, and this is the methylene. Then we have polyunsaturated fatty acid. That means I will have more than one carbon-carbon double bonds. An example of such a fatty acid is linoleic acid. And if you see linoleic acid, there are two double bonds. This is better explained in the molecular architecture of linoleic acid. You can see here, these are the two double bonds that are present, so more than one. So polyunsaturated fatty acid. This is the carboxylic acid functional group, and this is the methylene. So I believe that these structures will help you to understand the classification of fatty acid based on saturation. And these are very basic concepts. And if I give you a structure of a fatty acid in an exam, you should be able to classify them very easily by looking at the level of saturation and identify them if it is a saturated one or a monounsaturated one or a polyunsaturated one. Now we come to the most important concept here, which is known as the omega classification of fatty acid. Now, in order to understand, we have to understand two different concepts here. The first concept is, this is the functional group. The carbon next to the functional group is the alpha carbon, so this is the alpha end, and the methyl end, as we saw in the previous slide, this represents the omega carbon. So generally, when you are given structures of fatty acids, they will not give you the complete structure, but they will give you a structure something similar to this, and based on the position of the functional group, you should be able to classify the alpha carbon and the omega carbon. So let us start looking at the classification. So this is linoleic acid. Now if you look at it, they say this is an omega-6 polysaturated fatty acid. How do I know that this is a omega-6? So I know this is the omega carbon, so I start counting the number of carbons from the omega end. So 1, 2, 3, 4, 5 and 6. So I have encountered the first unsaturation from the omega end at the 6th carbon position. So that's why I call it an omega-6 polyunsaturated fatty acid, because I have two carbon-carbon double bonds present in linoleic acid. If I want to have this particular structure represented in the form of a numerical symbol, then I have to consider the counting from the alpha end. So this is linoleic acid, consists of 18 carbons. So this is an 18 carbon containing fatty acid, so 18. Then there are two unsaturations, so I write 18:2, and the position of the unsaturation now has to be classified from the alpha end. So 1, 2, 3, 4, 5, 6, 7, 8, and we have the 9th position where I get the first unsaturation. So I write delta 9, 10, 11, and then we have the 12th position where there is another unsaturation. So the numerical symbol for this particular fatty acid is 18:2, delta 9, 12. So when I am looking at the position of the double bonds in a numerical symbol, I start from the alpha end and go towards the omega end. When I am classifying it from the position of the omega carbon, then I look at the position of the first unsaturation from the omega end. And when I look at the first unsaturation from the omega end, I encounter it at the 6th position. So therefore I call it an omega-6 polyunsaturated fatty acid. Let's take another example. Arachidonic acid. Arachidonic acid consists of 20 carbons. So if I want to call it, classify it from the omega side, this is 1, 2, 3, 4, 5, and since I encountered the first unsaturation at the 6th position, This is an omega-6 fatty acid. Since there are more than one double bond in this particular molecule, I call it omega-6 polyunsaturated fatty acid. Now if I want to represent this particular molecule in terms of numerical symbol, then I start counting from the alpha side. So I have 1, 2, 3, 4, and I encounter the first unsaturation at the 5th position. So 20 is to 4 because there are 4 unsaturations. Then 5 is the first position where the unsaturation is present. Then 6, 7, and 8 is the second position where there is an unsaturation present. So I write 5, 8. Then I keep on counting 9, 10, 11. So I again encounter an unsaturation at the 11th position. So I write 11 here, and then I continue 12, 13, 14, where there is another unsaturation, and therefore I write 14 here. So this is an omega-6 polyunsaturated fatty acid. Numerically, it will be 20:4 delta 5, 8, 11, 14 fatty acid. So when we are looking at the numerical classification, we start from the alpha end, and when we look at the omega classification, we start from the omega end, and in the place where we encounter the first unsaturation is how the omega classification goes. So here it is omega-6, and since there are 1, 2, 3, 4, more than one unsaturation, I call it an omega-6 polyunsaturated fatty acid. Similarly, if you look at this particular fatty acid EPA, then omega-1, 2, 3. So this is a omega-3, and since there are more than one unsaturation present, I call it omega-3 polyunsaturated fatty acid. So based on what kind of classification we are looking at, we can classify fatty acids into 2 categories. First, numerically, and second, from the omega side. And this concept should be very clear, because in the exam you will expect at least one particular MCQ where the structure of the fatty acid will be provided to you, and you have to classify it either numerically or based on the omega classification. Now, as we move on, we talk about a specific class of molecules in relation to fatty acids, and these are known as triglyceride. Triglycerides are often abbreviated as triacylglycerol, or sometimes in many textbooks they write it as TAG. TAG stands for triacylglycerol. TG stands for triglyceride. In triglyceride, we have one glycerol molecule. So this is glycerol in this particular part, you can see here. So glycerol is a trihydric alcohol, and this glycerol forms a backbone where it forms ester linkages with 3 fatty acids. So this is one fatty acid, this is second fatty acid, and this is the third fatty acid. So triacyl, because there are 3 fatty acids, triacylglycerol or TAG or triglyceride. Now this particular fatty acids that are attached to the glycerol molecule may be of different kinds. What I want to say here is, for example, fatty acid 1 may be a saturated fatty acid, whereas fatty acid 2 may be a monounsaturated fatty acid, whereas fatty acid 3 may be a polyunsaturated fatty acid. Similarly, fatty acid 1 can be a short chain fatty acid, whereas fatty acid 2 can be a long chain fatty acid, whereas fatty acid 3 may be a medium chain length fatty acid. So most triacylglycerol or triglyceride contain more than one type of fatty acid, and this concept should be very clear, because this is how triglycerides help in the maintenance of lipid homeostasis. And this particular part, as I said, will be dealt in detail when we talk about lipoprotein metabolism in lectures concerning cardiovascular biochemistry. Now, if you want to identify this reaction, so this is a dehydration reaction that is happening. You lose 3 water molecules. So you have, this is glycerol, this is a trihydric alcohol. You can see here the 3 alcohol functional groups, and this reacts with 3 fatty acids, and each of these reaction centers forms an ester linkage, and you get a triglyceride or triacylglycerol molecule. In the body, triglyceride undergoes hydrolysis in the presence of an hydrolase, lipase, and this leads to the formation of 3 fatty acids, so 1, 2, and 3, along with glycerol. So triglyceride, sorry for, you should write here triglyceride, that's a typographical error. So triglyceride plus water in the presence of the enzyme lipase, which is a hydrolase, leads to the formation of 3 fatty acids and glycerol. Remember one thing, that when there is hydrolysis happening, the hydrolysis will first occur on these 2 fatty acyl groups, that means here, and after that this particular ester linkage will be hydrolyzed. That's because this ester linkage is not accessible to the enzyme for cleavage until and unless these 2 are cleaved. So a concept of steric hindrance comes into play here as well. So triglyceride plus water in the presence of the enzyme lipase leads to the formation of 3 fatty acids and glycerol. These 3 fatty acids can be of, most of the time will be of different kinds, as I explained to you in the previous slide. So summing it up, we have triacylglycerol or triglycerides, which undergo lipolysis in the presence of the enzyme lipase and water, leading to the formation of fatty acids and glycerol. Now our primary focus of this lecture is to realize what is the fate of these fatty acids and glycerol. So we start first with glycerol. What is the fate of glycerol? So this is glycerol that is derived from lipolysis. In the first step, glycerol is phosphorylated using ATP to use glycerol 3-phosphate. If I am using ATP, it will be a kinase that will be catalyzing the reaction. Since the substrate is glycerol, the enzyme is glycerol kinase, ATP is used in the process and you get glycerol 3-phosphate. In the second step, glycerol 3-phosphate is oxidized because you have a proton that is coming out. In the presence of an oxidoreductase, glycerol 3-phosphate dehydrogenase, and this glycerol 3-phosphate is then converted to dihydroxyacetone phosphate. Of course, dihydroxyacetone phosphate is a glycolytic intermediate, so this particular metabolite goes into glycolysis, and depending on the state of the cell, it can either undergo aerobic or anaerobic respiration. Also, dihydroxyacetone phosphate being an glycolytic intermediate can also be converted to glucose by the process of gluconeogenesis, which essentially is the reverse of glycolysis. So, to sum it up, glycerol in two steps. First is an ATP requiring step is converted to glycerol 3-phosphate, and in the second step it is oxidized to dihydroxyacetone phosphate, which is a glycolytic intermediate, and depending on the energy need of the cell can be converted either to pyruvate or glucose by glycolysis and gluconeogenesis respectively. Now this particular process, please remember this, happens only in the hepatocytes, which are able to convert glycerol to dihydroxyacetone phosphate. And as I mentioned, and I'm mentioning this over and over again, depending on the energy need of the cell, this dihydroxyacetone phosphate may either be converted to pyruvate to release ATP, or it may be used to synthesize glucose by the process of gluconeogenesis. So, to sum it up, glycerol derived from lipolysis in a two-step process is converted to dihydroxyacetone phosphate. Dihydroxyacetone phosphate, depending on the energy need of the cell, may undergo glycolysis or gluconeogenesis. Will I need to remember these two steps? The answer is yes, you need to remember these two steps because these two steps are often asked with regards to various inhibitor activity, and especially they are very important when you look at integration of metabolism. And of course, I believe Prof. Riyad will discuss this in detail when he talks about integration of metabolism towards the end of the course. So we have metabolized glycerol, but we are still remaining with the 3 fatty acids. So what is the fate of fatty acid? Fatty acids undergo a process called beta oxidation, and this beta oxidation leads to the production of acetyl coenzyme A. And I think you already know that fatty acids after beta oxidation lead to the production of acetyl coenzyme A. This acetyl coenzyme A, if there are enough oxaloacetate molecules, then will go into the TCA cycle to produce ATP through the electron transport chain. However, if oxaloacetate molecules are lacking, then this acetyl coenzyme A will go and form ketone bodies. And we have to look at this particular process in detail. This particular process you have already looked at in TCA cycle. Now we come to the different phases of beta oxidation. Of course, remember in the textbook or in other material you will not find this classification of beta oxidation into 2 phases. But I like to teach it in 2 phases because it is easy to understand and comprehend the process of beta oxidation. If we divide beta oxidation into first the activation phase, which mostly deals with the transport of the fatty acid molecule inside the mitochondrial matrix, and the second phase is the actual set of reactions that oxidizes the fatty acid. Now, before I start with the activation phase, you need to note down a couple of things. First, the site for beta oxidation is the mitochondrial matrix. Therefore, cells that lack mitochondria will not be able to pursue beta oxidation. For example, red blood cells or erythrocytes cannot metabolize fatty acids because they do not contain mitochondria. So at the end of lipolysis, I have fatty acid which is present in the cytosol along with glycerol. I have already metabolized glycerol into dihydroxyacetone phosphate, but this fatty acid, in order to be metabolized further, needs to come inside the mitochondria. So in the first step, in the presence of the enzyme thiokinase, fatty acid combines with coenzyme A. In the presence of ATP, leads to the formation of fatty acid or fatty acyl coenzyme A. In the process, I can say that I have used 2 ATP molecules. The reason, go back to your concepts that we talked about. I am breaking 2 high energy phosphate bonds in ATP, because if I look into this reaction, I am converting ATP to AMP. So this is adenosine triphosphate and this is adenosine monophosphate. So 2 high energy phosphate bonds are broken. Therefore, I have used 2 ATP to convert fatty acid to fatty acyl coenzyme A. The mitochondria, as Dr. Nerissa has already told you, has 2 membranes. So this is the inner membrane, this is the outer membrane. And at the end of this reaction, the fatty acyl coenzyme A migrates into the intermembrane space, which is the space between the inner and the outer membrane. So this particular space is called the intermembrane space. So this fatty acyl coenzyme A comes into the intermembrane space and undergoes an exchange reaction with a particular molecule known as carnitine. So this coenzyme A goes back out into the cytosol and will combine with further fatty acid to form fatty acyl coenzyme A. This carnitine molecule combines with fatty acyl, so we have fatty acyl carnitine that is formed in this particular step. Let us look at this in a bit more detail. So fatty acyl coenzyme A comes inside the intermembrane space, undergoes an exchange reaction with carnitine, coenzyme A is released and fatty acyl carnitine is formed, and this particular reaction is catalyzed by carnitine acyl transferase 1. So at the end of this step, we have fatty acyl carnitine. The fatty acyl carnitine now has to go inside through the inner membrane inside the mitochondrial matrix. So there is a particular enzyme called translocase which helps in the migration of this fatty acyl carnitine from the intermembrane space inside the mitochondria into the matrix. So formation of fatty acyl carnitine by carnitine acyl transferase 1, the enzyme, and once fatty acyl carnitine is formed, with the help of the enzyme translocase, it comes inside the matrix, and once it is inside the matrix, then there is a third set of reaction that will happen, and in the third set of reaction, it undergoes a second exchange where it exchanges carnitine with coenzyme A. Now this coenzyme A is present inside the mitochondrial matrix. In the process, carnitine is released, so carnitine goes out into the intermembrane space, and we have fatty acyl coenzyme A, which is the precursor molecule for the process of beta oxidation to start. So let us look at this process again. So fatty acyl carnitine comes through the inner mitochondrial membrane helped by the enzyme translocase, which we looked at it in the previous slide. Sorry about the numerous clicks, because otherwise I will not be able to write this. So with the help of the translocase, the fatty acyl carnitine comes through the inner mitochondrial membrane inside the matrix. Once the fatty acyl carnitine is inside the matrix, it undergoes a second exchange where it combines with coenzyme A. So at the end we have fatty acyl coenzyme A. The carnitine that is released in the process goes back to the intermembrane space, where it helps in the transport of other fatty acyl coenzyme A that is coming from the cytosol through the outer membrane. So this carnitine is coming here, and after coming here through the exchange reaction it is released, and once it is released it goes back into the intermembrane space. So this particular process where the carnitine is shuttling between the intermembrane space and the mitochondrial matrix is known as the carnitine shuttle. So the carnitine shuttle helps in the migration of fatty acyl carnitine or fatty acids from the intermembrane space into the mitochondrial matrix. And this concept should be very clear, and is very important, because not only these steps are clinically relevant, but numerous questions are asked in any exam in relation to these steps. And that's the reason I took some time to dwell on the reactions and the enzymes that are helping in the process. Do you need to remember them? The answer is yes, you need to remember all the 3 steps that we talked about. So now at the end of activation phase, we have been able to transfer the fatty acid from the cytosol inside the mitochondria, and therefore the oxidation phase will now start. In the activation phase, we were able to transport the fatty acid from the cytosol into the mitochondria with the help of the carnitine shuttle. So this is the acyl coenzyme A molecule, so fatty acyl coenzyme A. So this is the alpha carbon because this is the functional group, and this is the beta carbon. And look here, the process is called beta oxidation. So something is going to happen at this particular position, or more precisely, there will be an oxidation happening at the beta carbon, and that's the reason this particular reaction mechanism of metabolism is known as beta oxidation of fatty acid. So in the first reaction, we have a particular oxidoreductase called acyl coenzyme A dehydrogenase, which removes hydrogen from the alpha carbon, and you can see here, once the hydrogen is removed, you get a double bond that is formed between the alpha and the beta carbon. So this is the double bond that is formed. The proton that is released from the alpha carbon is trapped up by FAD, which forms FADH2. This FADH2, of course, will go to the electron transport chain, and this will lead to the production of ATP. So acyl CoA dehydrogenase catalyzes the oxidation at alpha position. In the process of oxidation, the released proton is trapped up or taken up by FAD, which is converted to FADH2. FADH2 migrates to the electron transport chain and leads to the production of ATP, similar to the TCA cycle concept. What is the second reaction that is happening? So we have the first reaction, acyl CoA dehydrogenase, where we lost one proton and we formed FADH2, and the end product was a trans-NOL coenzyme A. Trans because this particular hydrogen are in the trans orientation. So trans-NOL coenzyme A. And in this particular reaction, we have a hydration. So we use a hydratase And in the process of this, what happens, as you can see here, it forms a hydroxyl group at the beta position. So this is the alpha, this is the beta position, and you have a hydroxyl group that is formed because of this hydration at the beta position. Now we get beta hydroxyacyl coenzyme A, beta hydroxyacyl coenzyme A. So that's the reason we need a hydratase which will help in the process of hydration. Now we move on to the third step, and here again we have an oxidation that is happening, and if you look at it, this oxidizes the hydroxyl group. So oxidation at the beta position, so this is the alpha, this is the beta position, loss of protons, so these two protons are lost, and in the process NAD traps these protons and forms NADH, and this NADH again goes to the electron transport chain, and through a series of exchange reactions leads to the production of ATP, and you get a ketone group at the beta position because of this oxidation. Because the ultimate oxidation is occurring at the beta position of the fatty acid, we call it the beta oxidation of fatty acid. In the process of two oxidation reactions, the protons that are released are trapped by FAD and NAD respectively, which go into the electron transport chain to produce ATP. Now in the fourth step and the ultimate step, what happens is that by splitting the bond between the alpha and the beta carbon, so we split the bond between the alpha and the beta carbon, and we get an acetyl coenzyme A molecule, and we get a shortened fatty acid, and this shortened fatty acid goes into further beta oxidation cycles. And this process will be clear to you as I take an example and talk about beta oxidation of fatty acid. So let us take a C14 fatty acid, or specifically known as myristic acid. So this is the mitochondrial matrix, this is the fatty acyl coenzyme A. So this is the alpha carbon, beta carbon, then there are 10 more carbons, so we get 14 more carbons here, and this is the methylene that you see here. So in the first step we have the oxidation at the alpha carbon that is occurring, and that leads to the formation of a double bond. The proton that is released is trapped by FAD to FADH2, and FADH2 goes to the electron transport chain. Then we have the hydration reaction that is occurring, and this is the hydroxyl group that is formed at the beta position. And then in the third reaction we lose another proton from the beta position, and we get a ketone group here, and this particular part will now undergo cleavage so that I can get an acetyl coenzyme A molecule from this particular part. And if I look at it, I get one acetyl coenzyme A molecule, and then I get a fatty acid which is now less by one particular carbon, two particular carbon. So from C14 I go down to C12, then in the second beta oxidation cycle from C12 I go down to C10. In the third one we go from C10 to C8. In the fourth one C8 to C6, and then in the fifth one C6 to C4, and in the sixth one, of course, we will go down to 2 acetyl coenzyme A molecules. The reason being, at the end we will have no more fatty acyl coenzyme A remaining. So at each step, if you see here, you get a fatty acyl coenzyme A which is less by 2 carbons, but at the last step we will get 2 separate acetyl coenzyme A molecule. So in miricetic acid, I have used 6 cycles, but I have produced 7 acetyl coenzyme A molecules. If I want to write it down, if I have 12 carbons, the number of acetyl coenzyme A molecules that I will produce is 6, 12 by 2, but the number of cycles will be number of acetyl coenzyme A molecules minus 1. If I have a 14 carbon containing fatty acid, then I will produce 7 acetyl coenzyme molecules. The number of cycles required will be 7 minus 1, 6. If I have 16 carbon containing fatty acid, the number of acetyl coenzyme A molecules that I will produce is 8. However, the number of beta oxidation cycles I will require to metabolize this fatty acid is 7. If I have 18, then I will produce 9 acetyl coenzyme A molecules, but I will take 9 minus 1, 8 beta oxidation cycles to completely metabolize this particular fatty acid. So what is the thumb rule here? The length of a fatty acid determines the number of oxidations and total number of acetyl coenzyme A group that will be produced. Now we need to look at how to calculate the number of ATP molecules produced from a certain length fatty acid. So activation of the fatty acid molecule requires 2 ATP. Remember, I mentioned that when we talked about the activation phase, that ATP is getting converted to AMP, so I am using 2 high energy phosphate bonds. Therefore, for calculation purposes, I am using 2 ATP molecules. One cycle of oxidation of fatty acid produces 3 ATP molecules from 1 NADH, and 1 FADH2 molecule produces 2 ATP from the electron transport chain. So this is what you should use all the time for calculation of ATP in the exam. So I mentioned this before. 1 NADH will give rise to 3 ATP molecules, 1 FADH2 will give rise to 2 ATP molecules, and 1 acetyl coenzyme A on entering the citric acid cycle produces 12 ATP molecules. So this is like a rule book that you should remember, because you will definitely be asked a question on calculating the total or net gain of ATP from a certain chain length fatty acid. I repeat here, activation takes 2 ATP. One cycle produces 1 NADH that will produce 3 ATP molecules, 1 FADH2 that will produce 2 ATP molecule by the participation of the electron transport chain, and 1 acetyl coenzyme A molecule on entering the TCA or the citric acid cycle leads to the production of 12 ATP molecules. So let's take the example of lauric acid. I just took an arbitrary fatty acid. Lauric acid contains 12 carbons. So 12 carbon containing fatty acid will lead to the production of 6 acetyl coenzyme A molecules, and the number of cycles that I will require to metabolize lauric acid completely is 5. So activation of lauric acid will take 2 ATP molecules, and from 6 acetyl coenzyme A molecules, 12 into 6, I will produce 72 ATP molecules. 5 oxidation cycles will give rise to 5 NADH and 5 FADH2. In total, from these two to the electron transport chain, I will get 25 ATP molecules. So if I add 72 to 25, then the total number of ATP molecules that are produced is 97. However, I have used 2 ATP in the activation phase, so the net gain of ATP in the process will be 95. So I will cross this not to confuse you. and write here net gain. Total, of course, is 97. 72 plus 25. So before I come to this particular concept, I go back again, try to solve this with any number of carbon containing fatty acid. So you can start with a carbon with a fatty acid that is containing 18 carbons, and here you have to just substitute 12 with 18, and 18 into 6, 6 8s are 48, and therefore there will be 108 ATP molecules produced. But remember, I made a mistake here, because when I am looking at the number of carbon in a fatty acid, I also need to take into consideration the number of acetyl coenzyme A molecules that will be produced. So in a 18 carbon containing fatty acid, I will require 9, or I will produce 9 acetyl coenzyme A molecules, and I will require 9 minus 1, 8 oxidation cycles. However, these numbers change, but this particular part will not change. So whatever the number of carbons, I will always require only 2 ATP molecules for the activation phase, where I transport the fatty acid from the cytosol inside the mitochondrial matrix. So even this is 18 carbon, I will still use 2 ATP molecules. So if I want to write this down, so there will be minus 2 for the activation phase. Don't make this mistake that I was trying to highlight, that always take 6. So in this case I have 9, and 9 into 18, and 8 into 3, 8 into 2. You need to add all this up and subtract 2 from it, and you will get the net gain of ATP for a 18 carbon containing fatty acid. Of course, the total number will be without subtracting the high energy phosphate bonds that I have used in the process of activation. So try to practice this using different fatty acid with regards to different lengths or different carbon number with regards to fatty acids containing different number of carbons, and you will get the idea. But remember, whatever the number of carbons, I will always require 2 ATP molecules for the activation phase. But pay attention to these two numbers. People always make this mistake, that first you need to calculate the number of acetyl coenzyme A molecules and also the number of oxidation cycles. And this is my bad. We should have 12 here, sorry. So 12 ATP so per acetyl coenzyme A, so it will be 12 into 9, 108. So this is how you should calculate the number of ATP molecules produced from a given number or given fatty acid. So at the end, we have taken fatty acid, converted it to acetyl coenzyme A. A lot of acetyl coenzyme A is produced, as you can see, and this acetyl coenzyme A, depending on the availability of oxaloacetate, please mark this. Many of you made this mistake in the midterm assessment. Depending on the availability of oxaloacetate, will move into the TCA cycle. If oxaloacetate is not available, of course, this particular acetyl coenzyme A molecule will lead to the production of ketone bodies. Now we come to fatty acid oxidation disorders. Please, please, I'm asking you to go through this because I will be asking question on this disorder. I gave you a practice assignment for that. What you should do is to download the paper from the LMS, refer only to the highlighted parts. You don't need to read all the you do not have to really go through all the material in the paper, but only the highlighted parts, and that will give you a fair idea about fatty acid oxidation disorders. So fatty acid oxidation disorders, sometimes known as fatty oxidation disorders or FODs, are genetic metabolic deficiencies in which the body is unable to oxidize fatty acids to make energy because an enzyme in the process of beta oxidation is either missing or not working correctly. So either the enzyme may not be produced, or there may be a mutation in the gene which alters the structure of the enzyme. As a result, the person's enzyme or the patient's enzyme cannot perform the required function. The most important of this fatty acid oxidation disorder is the carnitine transporter deficiency. Carnitine transporter means carnitine in these people are absent, and this is an autosomal recessive disorder, so it's a very uncommon disorder. And in this particular disease, people are unable to metabolize fat because they cannot transfer the fatty acid inside the mitochondrial matrix. Of course, glycerol is produced. Glycerol goes into dihydroxyacetone phosphate, and that can be used for the synthesis of glucose by gluconeogenesis or for the production of ATP by glycolysis. Because this kind of disorders generally affect energy production, this affects the brain function, and these people have extreme insomnia or sleepless, extreme sleepiness, the reverse of insomnia, my bad, mood swings, poor appetite, and hypoglycemia. And the treatment for this is to administer L-carnitine. So carnitine transporter deficiency is one of the most well-studied fatty acid oxidation disorder. What you should do for me is to go through the marked or highlighted aspects of the review article and write or answer the questions that I have provided to you. It's a formative kind of assessment for you. If you do not want to do it, it's up to you, but I will be asking you questions in the exam from the sections that I have highlighted for the fatty acid oxidation disorders. So, we have talked a lot about carbohydrates. We converted carbohydrates to acetyl coenzyme A, citric acid cycle, carbon dioxide, and then we have the electron transport chain. Now today we talked about fats and fatty acids. Fatty acids or fat, we talked about the metabolism of triglyceride, which undergoes lipolysis to produce fatty acid and glycerol. Fatty acids go into the beta oxidation cycle to give rise to acetyl coenzyme A, which under normal physiological conditions should go into the citric acid cycle, leading to the production of high energy electron carriers, which go into the electron transport chain, leading to the production of ATP. However, the entry of acetyl coenzyme A derived from fatty acids is subject to the availability of oxaloacetate. If oxaloacetate is absent, that acetyl coenzyme A then goes into the ketone body formation pathway. And this is what we are going to talk about in the next lecture. So that's it for you. I will end the lecture here. Please focus on the assignment. Please focus on the calculations. If you have any difficulty in understanding, we can revisit these concepts again in the class. Thank you very much. and have a nice day.